So the title of my talk is Emerging Mechanisms of Activation in Cancer, and also their linkage to neurodevelopmental disorders, which is something that we have taken recently, and we find, as I will discuss a little later, we discuss a fascinating connection. So here is a schematic map of the MAPK pathway. We can see RAS here at the center. We see proteins that activate it with the arrow, proteins that inhibit it, proteins that activated coming from the upstream signaling and downstream signaling. Such simplified diagrams are highly informative and provide a very useful overall picture. They take a huge amount of time and effort to construct. And I think they're very popular. I mean, lots of groups, lots of people use them, including our own group. At the same time, they are unable to capture the root cause of pathological alterations. Looking at such a map, we still do not understand why different isoforms of RAS, KRAS, KRAS for A, KRAS for B, NRAS, HRAS, etc., populate different tissues, different cell types, why different mutations have different consequences, and why do they show different preferred distributions in the different isoforms. Looking at such maps, we still do not understand. In such maps, there are no cell type specific data and no structures. So, for example, they do not help us understand how does the PI3K activation take place in the membrane. PI3K is extremely, what happened? Okay, PI3K is an extremely important enzyme. It's a lipid kinase. It phosphorylates the PIP2 to the PIP3 at the membrane. Here is the crystal structure of the PI3K, and it has two subunits, the P110. Here is the PI3K alpha isoform, so it has the PI10 alpha. It includes the kinase domain, and the P85, which is the regulatory subunit, and it's often called the inhibitory subunits. There is a hydrogen exchange and mass spec data, which suggests that the activation of the PI3K of the PI3K is via the interaction of the NSH2 domain with the C-terminal motif of an RTK, receptor tyrosine kinase, like, for example, insulin receptor, specifically with this phosphorylated motif at the C-terminal of the receptor, which leads to the breaking of the interaction or of the interface between the NSH2 and the helical domain. So here in green, the helical domain. This is the NSH2 domain right here, leading to conformational changes at the ISH2 here in blue or cyan, with the C2, here C2 domain, and the ABD movement. However, how these remote actions regulate the PI3K activation, and how is PI3K activated by the NSH2 release are pivotal questions that, looking at this, we are not able to understand. And we know rigid molecules cannot be regulated. Consequently, they cannot sustain life. So we asked, how is this PI3K lipid kinase activated? We took the crystal structure, and into it we modeled the PIP2. This is the signaling lipid, which is phosphorylated by the PI3K to PIP3 by the transfer of the gamma phosphate of the ATP to the tip of the PIP2. When we modeled it, we could see that the distance between the gamma phosphate of the ATP and the tip of PIP2 is 6 to 7 angstroms, which is much too far for a phosphoryl transfer. We know in active PI3K, the distance should be between 1 and 3 angstroms. So we asked, how is the lipid kinase activated? And we particularly were interested in this enzyme since we, as I'll come a little later when I give an overview of our direction, we are interested in the RAS signaling network. So we looked at the PI3K activation. We knew that the release of the NSH2 is responsible for PI3K activation, and that the basic residues in the activation loop are important for substrate binding. And here we can see the NSH2 here in red. Here is the helical domain to which with which it shares an interface, and here is the ATP pocket with the positively charged residues. And we are interested in the mechanism. Oh, I'm sorry, I went backwards instead of, I did not, yes, okay. So we are interested in the mechanism of the PI3K activation by the NSH2 release. And here we see the details. Here is the ATP. This is the activation loop in gray or in black. This is the ISH2. We see the long gray helices and the basic patch. So we took up the mechanism of the PI3K activation by the NSH2 release. We started by simulating the inactive PI3K. We see that it just fluctuates, no major movement of the NSH2 with respect to the helical domain. We then, to observe the conformational changes that are involved in activation, what we now do is remove the NSH2, because we know activation involves the opening of the NSH2 from the dissociation of the NSH2 from the helical domain. So now what we do is simply remove the NSH2 to mimic this, and we simulate. Now what we see, that the ISH2 domain does move. To quantitate it, what we do is we look at the angles and the distances in the inactive PI3K and the active PI3K without the NSH2. So here in the inactive, we look at the angle between the C2 and the kinase C lobe, and the C lobe of the kinase domain. Here we see an increase in the angle between the inactive state and the active state, and we look also at the distances between the C2 and the kinase domain, and here we see that the distance in the active state is larger, increases with respect to the inactive state. Now we put it on the membrane. We know that the activation is at the membrane. So here again, we have the NSH2 domain. We simulate it at the membrane. Still nothing much happens, just fluctuations. We then do what we did before. We remove the NSH2. And now we simulate it and we see the opening up and the binding at the membrane surface of the kinase domain, and this is the ISH2 right here. So what we observe is that the NSH2 release promotes the membrane interactions of the PI3K in an appropriate way for the catalysis. What is it? Okay, for the catalysis. Now to look at the details, here we have again, this is the inactive state on the left-hand side. This is the NSH2, the ISH2, and the kinase domain. On the right-hand side, we see the ATP and the basic patch. Here we can see again the details, the ATP and the basic patch on the activation loop. And now we see this is the distance in the inactive state with the NSH2. We remove the NSH2, we do the simulations, and we follow this distance. And what we see is that this distance shrinks. So here again, we see the ATP and we see the basic residues. We did not put the PIP2 into the simulations. To try and substantiate our result, what we do is we go to inevitably to the PDB and we assemble, we collect all the crystal structures of the isoforms of the PI3K, PI3K alpha, PI3K beta, PI3K gamma, and PI3K delta. We superimpose all the structures, compare the distances, the angles, etc. And here is what we observe, and we focus on the different on the different important regions. And what we are looking for is to obtain insights into the activation mechanism from the ISH2 and the activation loop. So here now is the superpositioning of the crystal structures. This is the ISH2 in the inactive state in gray, in the active state in red, and we see that the ISH2 moves upon PI3K activation. And here we now see the movement of the ISH2. Now, we were wondering, in the literature, there are many papers trying to set the criteria that differentiate between the active, or the features that differentiate between the active and inactive states of protein kinases, none between lipid kinases. It's not that they are less important to cell function. We assume that the reason is it's more difficult to do at the membrane surface, membrane interface. We try to do this based on what we have observed and the data that we have. So here is again the inactive state, looking at the activation loop. Here in gray, we see that it is in a collapsed state. In the active PI3K, in red, we see that it is in the extended state. As to the alpha helix in the kinase domain, in the inactive state, it is in the in conformation. In the active state, it is in the out conformation. So the conclusion is that the NSH2 release reduces the PIP2 distance to the ATP in the kinase domain at the membrane surface. And here we now look. This is what we started with, this question of the distance, which was much too large. Now in the simulations, we obtain a distance of 2 to 3 angstroms, which we obtain upon the NSH2 release. So what is then the mechanism upon NSH2 release? In the inactive PI3K, the PIP2 faces, but is distal to the gamma phosphate of the ATP with a distance over 6 angstrom, being much too far for phosphoryl transfer. Upon NSH2 release, the PIP2 get closer to the ATP through the conformational changes at the kinase C, at the C lobe of the kinase domain, and the distance shrinks to 2 to 3 angstroms. Now, PI3K and PTEN, and I'll come to PTEN in a little later, are two proteins with a very large number of mutations in cancer. In fact, they are the second and third most contain. They are the second and third. They have the second and third most frequent cancer mutations right after P53. So what we wanted to do is to look at the mechanisms of these oncogenic mutations. There are two reasons we were particularly interested in this. First, we wanted, of course, to see the mechanism of the mutations. And what we were looking to see is that if we are right in our mechanism of activation of the PI3K, then the mechanism of the activation by the oncogenic mutations should be similar, because it's our inherent thesis that the activation mechanisms of mutations mimic the native activation mechanisms going from the inactive to the active state, except that this under physiological conditions, it's regulated. In cancer mutations, they become constitutive, but the mechanism itself is unchanged. And second, if we are right in our mechanism, we should see that indeed the mutations follow the mechanisms that we observed and we believe that hold. So what we then do, we look at the key, at the hotspot mutations, these two right here. They are at the interface between the NSH2 and the helical domain, and this one, the 1047, which is in the kinase domain at the membrane surface. And there are several additional mutations, which are weaker mutations here, which we also look at. So these two are involved in NSH2 release, and this one in membrane interactions. This is what we observe. We then look at the literature, and we see that indeed Peter Vogt, already quite a number of years ago in 2007, suggested that the helical domain and kinase domain mutations induce gain of function by different mechanisms, which is exactly what we observe. And here we look exactly how. So here, we said the first two break the interfaces. This one increases the charge on the membrane surface, the binding surface, and what was not suggested before, it also promotes an extended activation loop, which now we know is a typical feature of the active state. What happened? Why? Okay, and we look at another mutation. This is a weaker mutation, and we also see that it promotes an extended activation loop. So here are all the mutations that we looked at. We looked at single mutations, as one can see, but also double mutations. We, through collaboration with a colleague, first he was in Sloan Kettering, now he is in Columbia, an experimentalist who has a lot of clinical data. So these are double mutations, and what they observed is that the double mutations have a stronger effect than the single mutations, and they are also inhibited by drugs to a stronger, they're more sensitive to drugs, and the results that we have also are in agreement with this. Next, we look at the RAF kinase, a protein kinase. And we look at RAF again, as in PI3K, we started here too, at the autoinhibited state of the B-Raf. We start in the absence of the 1433 dimer. Now, there is no crystal structure of the entire RAF kinase, which immediately suggests that it's quite flexible, which is not surprising. It's expected looking at the long linkers and the flexible termini. But simulations, of course, can observe the more populated states. So we simulate, we see indeed that there are multiple states. We obtain ensembles, and we look here at this is the RAS binding domain and the cysteine rich domain. RAS binding domain, as the name implies, binds the RAS protein when the RAS is in the active GTP bound state. The cysteine rich domain binds the membrane and the kinase domain. So we look then at the superimposed structures, and they indicate that the RBD and CRD can interact with the kinase domain here in the autoinhibited state. As we were simulating it, the cryo-EM structure of the B-Raf now was published, two groups, Michael Eck from Harvard and John Kurian. We started in their structures. The RBD was not there. There was some density, electron density. However, it was too flexible, and it sampled too much of the space. However, the simulations again, so I should say we started then the simulation again, this time with the 1433 dimer. And now we were able to obtain the structure and position the RBD where in agreement with the electron density. So here we have now the structure here at the bottom is the 1433. So what we observe then from the simulations is that the dimer, the 1433, blocks the RAF dimer interface, which is required to be formed for RAF's activation, and it covers the membrane binding region of the cysteine-rich domain, and of course, it stabilizes the autoinhibited state of the RAF of the B-Raf isoform, all of which explains why the 1433 is there. And here are the details and the two phosphorylation sites on RAF, which interact with the 1433, and the release of this one, this phosphorylated serine at position 365, and the RBD CRD then activates the RAF. What we do next, we try to create an overall picture of the release of the autoinhibition of RAF. We have two scenarios. One without the 1433, which we believe can take place under the conditions when there is a low population of the 1433 in the cell, and the second when there is a high population of the 1433. In both cases, the scenario is unchanged. We know that the RBD, the RAS binding domain, binds to the active RAS, GTP bound RAS, and the CRD at the membrane. And this occurs whether it is bound to 1433 or there is not, or whether it is not bound, it is free, or it is bound to the 1433. So what is the mechanism through which the auto-inhibited state, whether in the presence or absence of the 1433, gets activated? So what we suggested is the obvious, that there is in solution, the highly populated state is the auto-inhibited state, simply because it is more stable. However, even in the auto-inhibited state, there is a minor population of the open RAS-free state. Now, the driving force for the interaction of RAS with RAS binding domain is the very high affinity of the RAS binding domain to the active RAS. So those sparsely populated conformers of the open state, the high energy state, bind to RAS. They get stabilized, and there is a shift of the ensemble to the more stable RAS-bound state. So our premise is that there are multiple B-Raf conformations in solution. Most are closed because this is the more stable state, but some are open, and with the high affinity, this is how they bind, and there is a shift of the ensemble toward the stabilized active state. So this we published in last year. Recently, Ping Zheng and Debbie Morrison published a paper also. So this is a very nice experimental paper. And in the paper, they also suggest a mechanism for the activation of RAF. In there, and this is of course a copy and paste from their paper, and the figure and also the legend, except that I tried to shrink it. It was several additional lines. It was too long. So in their scenarios, what happens is that there are steric clashes and electrostatic repulsion between RAS and the 1433 when the whole complex thrives or in the process of binding. This then results in these clashes in repulsion result in conformational changes, which then dislodge the RAS binding domain and the cysteine-rich domain from the 1433, leading to rearranging of the 1433 dimer binding, which in turn leads to exposing that key phosphorylation site and the RAF dimer interface. The CRD then rotates to interact with the membrane and RAS to further stabilize the interaction. And the exposed kinase domain dimerizes, and the active conformation is then stabilized by 1433. Obviously, this mechanistic outline is very different than what I have shown you. We believe that the key difference is in the view, which ours is physics-based. This mechanism is based on a single structure in solution. Our mechanism is based on viewing an ensemble of conformations. So if you have an ensemble of conformations, of course, different populations, according to the relative energies, we don't view it as these sequential changes to that single structure. So to me, this is an example of how important it is, which way one, the basic view or the basic grasp of what happens of protein structures and the behavior of proteins as ensembles versus single, as I said, conformations. So here is then the activation mechanism from based on the simulations, the cysteine-rich domain at the membrane, the RAS binding domain, which binds to activated RAS. So here in green, it is the GTP. RAS has to be GTP bound in order to get activated. RAS, of course, here is in a dimeric state. It doesn't have to be dimer. It can be clustered. So long as it's sufficiently close is okay. And here is the kinase domain of RAF, and it has to dimerize in order for activation of RAF to take place. The next question that we ask is how then RAF's side-to-side activation promotes the off-to-on transition. So here we looked, and what we suggested is that in the monomeric RAF, we have an intra molecular pi-pi stacking, which stabilizes it. This is replaced in dimerization by intermolecular pi-pi stacking at the interface, which releases then the helix, the alpha helix at the kinase domain, the C lobe of the kinase domain, which promotes then the off to on transition. Next, what we have done, I'm not going to go into it, is how RAF's dimerization then activates MEK, and the difference and why. So why RAF protein kinase activates MEK, and what happens to KSR, which is a pseudokinase. So that should be coming out in the cellular and molecular life sciences. And in a future time, I will discuss this as well. No time to discuss everything today. So to take an overall view of where we are going, our aim is to elucidate the mechanisms of key nodes in the oncogenic RAS signaling upstream and downstream. Major RAS pathways, signaling pathways that we have been looking at: MAPK involving RAS, RAF, MEK, ERK, KSR, and the PI3K, PDK1, AKT, mTOR, and PTEN, which is a tumor repressor. Activation at the membrane that we have been looking at is RAS, PI3K, PTEN, PDK1, AKT, RAF, and KSR. So these are at the these are also membrane attached, and the pathways, both of them consist of cascading phosphorylations by protein and lipid kinases and phosphatases. Phosphatases, here PTEN, all are major drug targets. And upstream nodes that we have been looking at so far, the source, the exchange factor of RAS, GDP by GTP, and GREB2, a scaffolding protein, and the BCR-ABL, of course, key protein in leukemia. So mechanistic questions in oncogenic RAS that we have been looking at so far include: how is RAS activated at the membrane, and how is frenesylated hypervariable region that can then regulate signaling? Why the differential statistics of its cell type specific isoform expression in distinct cancers? What we could not understand just looking at that simplified pathway diagram that I have shown you before. How to explain explain the perplexing two-state behavior of the KRAS4A isoform, which can behave both as KRAS4B and an NRAS. That's to say, it's active in both types of cancers that are populated by these other RAS isoforms. How to explain that? What are the mechanisms of specific oncogenic mutations and the inhibition? What are RAS dimeric states? What is the role of calmodulin and why only in KRAS4B? And what is the detailed mechanism of RAS activation? As to mechanistic questions on nodes in RAS signaling pathways that we have been looking at so far, these include RAF. In RAF, how is B-Raf auto-inhibited? How is B-Raf auto-inhibited in the presence and absence of 1433? And how is monomeric B-Raf, this particular mutation V600E, activated? And now we are also looking at V600K. What is the detailed mechanism of RAF dimer activation? As to MEK, what is the mechanism of MEK's activation by B-Raf and KSR? As to PI3K, lipid kinase, we have been asking related to the phosphorylations and the mutations in PI3K and in PTEN, and asking, are there also passenger phosphorylations? There are many phosphorylation sites in the PI3K and PTEN. Is it possible that some of these are just passenger phosphorylation, just like there are passenger mutations? Very simply, we know the kinases recognize the consensus sequences, and these are only several amino acids and they don't have to occur exactly. So is it possible that some places are not by passenger women are not functional in the cell? How do PI3K driver mutations work? What are the features that distinguish between the active and inactive PI3K lipid kinases? What is the mechanism of PI3K activation? As to PTEN phosphatase, what is the mechanism of activation of tumor suppressor PTEN at the membrane? So PI3K phosphorylates PIP2 to PIP3. P10 dephosphorylates PIP3 back to PIP2. So we looked at the P10 tumor suppression, which is at its localization in the cytoplasm and activation at the membrane. So the mechanism at the membrane of this pivotal phosphatase has been unknown. So here we see, this is what we call, this is the autoinhibited state of PTEN, which is what we called in the closed conformation. This is the C-terminal tail, it is in the phosphorylated state. This is the PIP2 binding domain, it is collapsed here. Dephosphorylation opens the tail, releases it. Now it translocates to the membrane and it interacts with the membrane. And we track the P10 from the Zwitter ionic membrane here onto the anionic membrane that harbors the PIP3. Here we see the PIP3. And observe that the PIP3 here allosterically unfolds the PIP2 binding domain, positioning it for interaction with PIP2. And then we determine the P10 catalytic action at the membrane. So here is the autoinhibited state of the phosphorylated PTEN in the double closed state, the tail, phosphorylated tail, and the folded PIP2 binding domain. And here now we looked at mutations of the PTEN, driver mutations, and interestingly, PIP10 and other proteins in these pathways as well can elicit both cancer and neurodevelopmental disorders. In P10's case, it is autism. So we became very interested in this scenario of mutations occurring in the same proteins and sometimes the same mutations leading to cancer and neurodevelopmental disorders. So here we looked at the P10 mutations in disease. Here you see mapped in the phosphatase domain. So here are mutations in P10 which relate to cancer. And here are mutations which relate to neurodevelopmental disorders. Specifically, it's called ASD, the autism spectrum disorder. And here we see these are those that lead to the autism spectrum. These are germline or the mutations, and these are the syndromes, and these are the mutations that lead to cancer, and these are somatic mutations. They occur largely during an individual's life. So, of course, we always want to understand why and how to understand. So we asked ourselves, how can same gene mutations and sometimes the same mutations promote both cancer and neurodevelopmental disorders? We know the phenotypes are very different between, consider cancer versus autism. So the symptoms of cancer are distinct from neurodevelopmental disorders, even though they may harbor the same gene and even the same mutations. So why mutations in the same gene can lead to such seemingly disparate phenotypes has been puzzling. It has also been puzzling why those with neurodevelopmental disorders have a higher risk of cancer. And examples, it's not only P10, RAS, RO, ERK, PI3K, P10, yes, and SHIP2 are among these oncogenic proteins as well as others. And the disorders include autism spectrum disorder, PI3K-related overgrowth spectrum of cerebral palsy, rasopathies, and more. So we looked at this, and this came out in Scientific Advances in January. So how to link same gene mutations to neurodevelopmental disorder versus cancer has been enigmatic. Some publications focused on the developmental disorders and cell cycle, whereas others explored mutational properties. A decades-old attractive hypothesis argued that the primary feature distinguishing between the two types of mutations is signaling strength and duration. If strong and short, developmental disorders. If weak and sustained, the likely outcome is cancer. This hypothesis has been tested by phosphorylation levels for MAPK associated rasopathies. However, the linkage between a mutation and signal strength and duration and the differential clinical outcomes have been elusive, and also signal strength and duration are unlikely to be independent parameters. So Here we see somatic and embryonic mutations in cancer and neurodevelopmental disorders, here mapped on RAS. Here, these are somatic mutations in cancer. These are in embryonic mutations in RAS. It would be the RAS erasopathy. Here we see the strong G12 mutations, and I should say this is cancer, but just now, Kevin Shokat said that he is focusing on developing an inhibitor for G12S serine, which is a mutation. These are cancer, which is a mutation in rasopathy. So same mutation. So embryonic germline mutations may or may not be stronger. In RAS, they are not. So here we drew schematic diagrams of the networks and cell cycles of a wild type cell on the left, a cancer cell, and a mutant embryonic brain cell, because it's neurodevelopmental disorders. Here we see normal cell cycle progression in cancer. If the signaling is strong, there is a fast, of course, cell cycle progression. If, however, it is very strong, it leads to OIS, oncogene-induced senescence. This is in mutant embryonic cell. It leads to premature senescence. So we suggested that the signaling levels are critical in determining the disease consequences, and these are the outcome of mutation strength, expression levels, cell type, and the timing window. We know different cell types, of course, different chromatin accessibility of certain genes. Consequently, this would affect expression levels, and this depends, of course, timing window. We know the packaging of chromatin during development, or in different cell types, the expression levels of different genes and different isoforms in skin cells is different than that, say, in pancreatic cell. So here we looked again, here in this case, PI3K signaling in neurodevelopmental disorders in nerve cell, in somatic cell. This is for both, this is in cancer, PI3K and PTEN, and this is in PTEN signaling in brain cell in neurodevelopmental disorders. So we propose that signaling levels are critical in disease consequences, and they are the outcome of mutation strength, expression levels, cell type specificity, very important. So simulations cannot is not the end, of course, is only present a specific aspect, and the typing and the timing window, somatic versus embryonic or germline, chromatin accessibility for the specific gene and for other genes, encoding other nodes in the pathway. Signaling questions also include that we have been interested in. I'm not going to go into details because my time is up. For example, we asked, are the drug resistance pathways that emerge following targeting of common driver mutations also common among patients? For example, consider that two people or in two different tissues have the same cancer arising from the same mutation, for example, G12C. And they're treated by a certain drug, and there is a development of drug resistance. What we ask is, would the resistance pathways in one individual would be the same as in the other, even though the cancer is the same, and the original mutation in the cell is the same. And we suggested, this was published in also, I think, I can't remember, I think January or December. So we suggested that the responses to the drugs are likely to differ. And this is because the mutant protein may be in different cell or tissue types, suggesting, again, think of chromatin, the expression level of that particular mutant would be different, and of other proteins in the pathway would be different. The cell state may differ, that's to say, developmental state, etc., for example. So it would affect gene expression profile, abundance of other proteins in the pathway, and the identity and patterns of background mutations, which may or may not be expressed a priori, because we all have many other mutations that we don't know are there because they exist in only very rare population of cells. But once the other cells are killed by the drugs, then these cells proliferate. And we also asked how to measure then signaling. For example, this is very important, can we predict whether the signal that propagates from an upstream to downstream node activates transcription? And we suggested a signaling by numbers, where we looked at successful signal transduction by the number of active molecules, not just that are activated by the protein, so it's the strength of the mutation, and the signal as it propagates downstream, which depends on the level of expression levels of the other proteins. And here are the acknowledgements to my colleagues in the group at Koch University, Oslem, Attila, Serena, and now more additional students at Shanghai Jiao Tong University, University of Illinois, Vadim, a longtime colleague, in Frederick, Northeastern, University of Maryland, Belgium, Anna Sablina, I've not shown all, and more recently at the Cleveland Clinic, with especially Faixiong Cheng. And now I'll end with putting this again slide on the screen with our profound view that all conformations pre-exist, and activation, whether wild type or mutation, leads simply to a shift in the ensemble. Under normal conditions, the inactive state is more stable. Activation, for example, by mutations, shifts the equilibrium towards the active state. And with that, I shall thank you, and I'll be happy to, if you have any questions.